Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating and relatively recent discovery in the realm of neurobiology, the glymphatic system. For a long time, the central nervous system, the brain and spinal cord, were thought to lack a traditional lymphatic system, the network of vessels that helps clear waste and excess fluid from the rest of the body. However, scientists have now identified a unique system within the brain that performs analogous functions, crucial for maintaining a healthy internal environment. Throughout the body, the lymphatic system plays a vital role in clearing away metabolic byproducts and managing fluid balance. It's also integral to the body's immune responses. Given the brain's high metabolic activity and the need to protect its delicate tissues, the absence of a similar robust clearance mechanism was a long-standing puzzle. The discovery of the glymphatic system has shed light on how the brain tackles the essential task of waste removal and has opened up exciting new avenues for understanding brain health and disease. The glymphatic system essentially acts as the brain's housekeeper. It facilitates the bulk movement of cerebrospinal fluid, or CSF, from the subarachnoid space, which surrounds the brain, along the spaces surrounding arteries, known as periarterial spaces. Here, the CSF mixes with the interstitial fluid, the fluid that bathes the brain cells within the brain tissue itself, or parenchyma. This mixture then flows out of the parenchyma along the spaces surrounding veins, called perivenous spaces, eventually draining into the peripheral lymphatic system. This directional flow, from arteries to veins, is crucial for clearing waste products from the brain tissue. The concept of fluid movement within the brain is not entirely new. Scientists had observed the entry of CSF into periarterial spaces and its mixing with interstitial fluid in earlier studies. The idea that this pathway might be involved in waste removal also had historical precedence. However, the unique aspects of the glymphatic system's discovery lie in several key features. Firstly, the directional nature of the fluid transport was established, with CSF entering along arteries and exiting along veins. Secondly, it was demonstrated that a significant metabolic waste product, amyloid beta, a protein implicated in Alzheimer's disease, is exported via this glymphatic pathway. Thirdly, the process relies heavily on the polarized expression of a water channel protein called aquaporin-4, or AQP4, which is found on the end feet of astrocytes, star-shaped brain cells that surround blood vessels. Finally, research revealed that this fluid transport and the entry of CSF into the brain tissue are strikingly increased during sleep, coinciding with enhanced clearance of metabolic waste. This observation has led to the hypothesis that the restorative function of sleep is partly due to the glymphatic system actively clearing the brain of toxins that accumulate during wakefulness. Since its initial description in 2012, the glymphatic system has sparked considerable interest and some debate within the scientific community. One of the main challenges has been the difficulty in studying this low-pressure fluid transport system non-invasively in a living, active brain encased within the rigid skull. The very procedures used to investigate it, such as injecting tracer molecules after opening the skull, can inadvertently disrupt the normal function of the glymphatic system. Furthermore, the redistribution of these tracer molecules after death can complicate the interpretation of histological analyses aimed at mapping fluid transport pathways. The understanding of fluid movement in biological tissues in general has also evolved, with the recent discovery of previously unrecognized fluid compartments that can collapse during tissue preparation, highlighting the need for in vivo imaging techniques. Additionally, the recognition that glymphatic activity is significantly influenced by brain states like sleep and wakefulness, as well as anesthesia and the time of day, underscores the importance of considering these factors when comparing different studies. Given the ongoing exploration of the glymphatic system, this review focuses on some of the fundamental questions that remain unanswered. These include the basic necessity of a dedicated fluid transport system for the brain and the many unexplored facets of brain fluid transport. The review also presents evidence suggesting a complex interplay between the glymphatic system and other bodily systems, including the cardiovascular system, the gastrointestinal tract, and the traditional lymphatic system. The authors propose that this interconnected network may act as a crucial communication axis between the brain and the rest of the body. To better understand the unique aspects of the glymphatic system, it's helpful to compare it with the more familiar lymphatic system found throughout the body. Both systems are essential for maintaining tissue balance, facilitating the movement of interstitial fluid, supporting immune functions, and clearing waste products. In peripheral tissues, blood capillaries continuously supply an ultrafiltrate of plasma. This fluid circulates around cells, delivering nutrients and removing waste, eventually draining into lymphatic vessels that return the fluid to the venous circulation. In the brain, however, the blood-brain barrier, a highly selective barrier, restricts the ultrafiltration of plasma in most areas. To compensate for this, the central nervous system produces its own fluid, the CSF, primarily in the choroid plexus, a specialized tissue lining the brain's ventricles. Some CSF production might also occur via the influx of plasma across the extensive network of brain microvessels. After its production, CSF circulates, in part, into the brain tissue along the periarterial spaces, forming the entry point of the glymphatic system. Thus, in both peripheral tissues and the brain, fluid entry into the tissue occurs at the arterial end of the microvascular network, likely driven by the pulsatility of arteries that propels fluid inward. However, the exit routes for interstitial fluid differ. While lymphatic capillaries are the primary drainage pathway in the periphery, in the brain, interstitial fluid exits along the perivenous spaces and cranial and spinal nerves. Eventually, CSF and interstitial fluid drain from the central nervous system via a traditional lymphatic network located in the meninges, the membranes covering the brain. Fluid also drains along nerve sheaths in the cribriform plate, a bony structure at the base of the skull, leading to cervical lymphatic vessels. Both these pathways ultimately lead to the superficial and deep cervical lymph nodes. All CSF eventually returns to the venous circulatory system, either indirectly through the lymphatic system or directly via structures called arachnoid villi. This indicates that interstitial fluid transport is directional in both the body and the brain, with distinct entry and exit routes. Unlike lymphatic vessels, which possess valves to prevent backflow, it remains an open question whether the glymphatic system has analogous one-way mechanisms within the brain tissue to ensure directional fluid movement. The glymphatic system is known to clear various substances from the brain, including potassium ions, waste metabolites like lactate, and peptides and proteins such as amyloid beta. Studies have shown that the rate at which tracer molecules injected into the brain tissue are clear depends on the injection site, suggesting that there might be regional variations in waste clearance within the brain. Supporting this idea, studies in mouse models of Alzheimer's disease have found reduced CSF and interstitial fluid exchange in certain brain regions compared to others, which correlated with the deposition of specific proteins in those areas. These findings in the brain have parallels in other organs like the gastrointestinal tract, where the composition of lymph and immune function can differ between different anatomical regions. Furthermore, the efficiency of glymphatic clearance is not constant. It varies with sleep state and time of day, being increased during both sleep and the inactive phase. This complexity suggests that the mechanisms and routes of fluid exit from the brain are more intricate than a simple plumbing system. Despite the progress in understanding the glymphatic system, many fundamental questions remain. We still lack precise knowledge of where interstitial fluid moves within the brain and whether there are areas of stagnant or slow flow. The exact points and mechanisms by which the glymphatic system connects to the traditional lymphatic system are also yet to be fully elucidated. Our current understanding of the anatomical pathways of interstitial fluid clearance in the brain is largely based on experimental procedures involving invasive intracranial injections, sampling at specific time points, and observing the bulk transfer of tracer molecules. The perivascular space, a key component of the glymphatic system, is distinct from the complex interstitial space of the brain parenchyma. It surrounds the brain's blood vessels and is lined by the end feet of astrocytes, which are closely associated with pericytes and the endothelial cells forming the blood-brain barrier. Estimates of the extent to which astrocytes cover capillaries vary, possibly due to the effects of post-mortem tissue processing and the limitations of microscopy techniques. Detailed three-dimensional electron microscopy has shown complete coverage of some capillaries by astrocyte end feet, while other studies using different fixation methods have reported less complete coverage. Recent research has indicated a stronger correlation between the size of astrocyte end feet and the diameter of arteries compared to veins, although these observations were limited to specific types of blood vessels in the cerebral cortex. However, the thickness of the astrocyte ensheathment around arteries, capillaries, and veins appears to be relatively consistent. Beyond these studies focused on the cortex, there is limited detailed information about astrocyte and foot coverage of arterial or venous walls and how this relates to the perivascular space. This lack of precise anatomical data directly limits the accuracy of models trying to simulate glymphatic fluid flow, where the geometry of the perivascular space is a critical factor in determining the calculated rate of CSF influx into the brain tissue. Acute changes in the geometry of the perivascular space caused by vasoconstriction, vasodilation, or the swelling of astrocytes have the potential to significantly affect the movement of glymphatic fluid. Evidence supporting this comes from studies in animal models of disease. For instance, the constriction of blood vessels widens the perivascular space, facilitating a rapid influx of CSF into the brain parenchyma. Conversely, during cortical spreading depression, a wave of neuronal depolarization linked to migraine aura, the perivascular space constricts, leading to reduced interstitial clearance of tracer molecules. Spreading depression involves an initial rapid vasodilation followed by vasoconstriction. It has been suggested that the vasoconstriction phase is associated with the swelling of astrocyte end feet, which contributes to the reduction in fluid flow within the perivascular space. However, the exact degree of vascular reactivity or end foot swelling needed to disrupt glymphatic flow is still unknown. The perivascular and vascular compartments are dynamic spaces that may provide a mechanism for coupling increased blood flow with enhanced waste clearance in the brain. In humans, slow brain waves that occur during sleep are coordinated with hemodynamic oscillations, which are in turn linked to the flow of CSF. Specifically, a nocturnal increase in low-frequency neuronal activity triggers increased cerebral blood flow, which paradoxically reduces the amount of CSF movement in the brain's ventricles and tissue, supporting the idea of a dynamic switching between blood and CSF volumes. Acute high blood pressure can increase the stiffness of arterial walls, resulting in decreased pulsatility and a reduction in perivascular fluid flow by as much as 50%, highlighting the close relationship between the vasculature and the perivascular space in glymphatic function. Body posture has also been shown to influence glymphatic flow, but whether this is related to known changes in cerebral blood flow, intracranial pressure, or sympathetic nervous system activity requires further investigation. Given the crucial role of astrocytes in the neurovascular unit and their direct involvement in regulating cerebral blood flow, it is plausible that astrocytes also play a role in managing volume dynamics between the blood vessels and the perivascular spaces. Beyond the spatial organization of the perivascular space, the primary evidence for astrocytic regulation of glymphatic fluid movement is the fact that AQP4 facilitates this transport. Normally, AQP4 is highly concentrated on the plasma membrane of the astrocyte and feet facing the perivascular space. This localization is maintained by a protein complex called the dystrophin-associated complex. Mislocalization of AQP4 away from the astrocyte and feet has been consistently linked to impaired glymphatic function in various studies. While this polarized expression of AQP4 is a key regulatory mechanism, it is likely that astrocytes can influence glymphatic function through other means as well. It is evident that efficient glymphatic function relies on optimized perivascular spaces with minimal resistance to fluid flow. However, few studies have examined whether long-term changes in the shape, permeability, and openness of the perivascular space are associated with glymphatic dysfunction. With increasing age, glymphatic flow tends to decrease, accompanied by increased reactive gliosis, a response of glial cells to injury, and a reduction in the polarized expression of AQP4 on astrocyte and feet. Reactive gliosis, characterized by increased expression of a protein called GFAP and the mislocalization of AQP4, is a common feature of neuropathological conditions and likely contributes to decreased fluid flow. Structural changes in the brain's blood vessels also occur with age, including increased tortuosity or twisting, which likely impedes fluid flow. The structure and function of the cerebrovasculature also change in disease states. In cerebral amyloid angiopathy, amyloid beta protein accumulates in the walls of blood vessels, causing them to weaken and the perivascular space to collapse. In small vessel disease, often associated with high blood pressure and diabetes, the arteries stiffen and undergo remodeling, leading to an enlargement of the perivascular space. The functional consequences of this chronic remodeling of the perivascular space are still largely unknown, raising several important questions. For example, if the curvature and surface area of the vasculature increase, can astrocytes compensate by adjusting their coverage? Do changes in astrocyte volume or shape, such as those occurring during reactive gliosis, directly impact local glymphatic flow? And finally, can the boundaries of the perivascular space ever break down, and what would be the effect on directional fluid flow? Astrocytes are remarkably complex cells with numerous fine, irregular processes extending beyond their vascular end feet. These processes can exhibit localized calcium signaling independent of the main cell body and other processes. Astrocytes and their processes can also undergo rapid changes in volume within seconds. This process seems to be independent of AQP4 and can occur under conditions of high potassium ion concentrations, such as during increased neuronal activity. This is consistent with the reduced interstitial space observed during wakefulness and likely represents a key mechanism for rapid adjustments in glymphatic flow. More gradual changes in astrocyte morphology have been observed in brain regions like the hypothalamus. Astrocytes can extend and retract their fine processes depending on sleep states, hydration levels, pregnancy, lactation, and the light-dark cycle. These morphological changes can last from hours to days and are generally reversible. For instance, during lactation, glial processes retract from a specific region in the hypothalamus and remain retracted until the pups are weaned, at which point they extend back into the area. It is currently unknown whether these astrocytic morphological changes influence glymphatic function. The brain's intricate neuronal networks, responsible for complex behaviors, tasks, and thoughts, rely on tightly controlled concentrations of ions in the interstitial fluid for proper signaling. Even subtle changes in interstitial ion concentrations can affect arousal states, with lower levels of certain ions and higher levels of others associated with increased brain activity. Astrocytes are highly dynamic regulators of interstitial potassium ion concentrations and pH, and they likely influence the levels of other ions as well as the volume of the interstitial space. It is currently unclear whether the glymphatic system itself independently contributes to the regulation of interstitial ion concentrations. However, there is reason to believe that fluid transport and the ionic composition of the interstitial fluid are closely intertwined. The strongest evidence linking brain-wide neuronal activity and glymphatic activity comes from sleep research. Non-rapid eye movement, or NREM, sleep is characterized by distinct patterns of brain electrical activity, including slow oscillations in the low-frequency range of 1 to 4 Hz. There is strong evidence that glymphatic flow increases during NREM sleep, facilitating the delivery of larger volumes of CSF to the brain. Similar increases in glymphatic influx have been observed under anesthetic regimens that also induce prominent low-frequency brain activity. In humans, the movement of CSF within a specific brain ventricle is also increased during NREM sleep, and genetic variations affecting AQP4, the water channel, are associated with changes in the architecture of NREM sleep, suggesting a connection between fluid movement within the brain and neuronal activity. During REM sleep, the brain exhibits low-voltage, rapid, desynchronized neuronal activity that superficially resembles wakefulness. Whether glymphatic flow decreases during REM sleep, as it does during waking, remains to be determined. The mechanisms underlying the functional relationship between neuronal activity and bulk fluid movement are still largely unexplored, and the impact of sleep deprivation or insomnia on these processes also needs investigation. Synchronizing the activity of distinct populations of neurons might influence local blood flow and fluid dynamics, ultimately altering the transport of solutes in specific brain regions. While this has been shown for CSF dynamics across the entire brain, it has yet to be demonstrated for localized waste clearance in particular brain areas or around individual blood vessels. Sensory stimulation targeted to specific neural networks, which are often impaired in aging and neurodegenerative conditions, such as high-frequency gamma oscillations seen during responses to external stimuli, might potentially improve glymphatic clearance. Support for this hypothesis comes from studies in mouse models of Alzheimer's disease, where stimulating sensory systems could alter local waste buildup. For example, inducing synchronous gamma oscillations using flickering light has been reported to reduce amyloid burden in the visual cortex. Similarly, entraining the auditory system with specific tones led to decreased amyloid protein accumulation throughout the cortex via mechanisms involving glial cells and blood vessels. While waste clearance is likely a primary function of the glymphatic system, it may also serve additional roles, including the widespread distribution of signaling molecules within the brain. CSF itself might play a significant, though perhaps underappreciated, role in transporting central nervous system signaling molecules to peripheral tissues. CSF is primarily produced by the choroid plexus located in the brain's ventricles. From there, it follows a directional path, flowing from the lateral ventricles to the third ventricle, through the aqueduct of Sylvius, and finally to the fourth ventricle, where it exits the brain via specific openings. From these openings, CSF enters a large cistern at the base of the brain and is either shunted out of the central nervous system via the meningeal lymphatic system or redirected back into the brain by the glymphatic system. The entry point for the glymphatic system is via another basal cistern, where CSF moves along the perivascular space surrounding a major artery to a key vascular structure at the base of the brain. From there, CSF ascends along the perivascular spaces of the major cerebral arteries. Studies in rodents have shown that CSF transport along the glymphatic system is rapid during sleep, taking only minutes to hours. In contrast, during the active phase, CSF is redistributed to lymph nodes in the jaw area with similar speed. The choroid plexus secretes or transfers a variety of substances from the blood into the CSF, including solutes, metabolites, vitamins, and carrier proteins. Interestingly, many brain regions that produce signaling molecules capable of long-range communication are located near the ventricular pathway of CSF transport. Classical neuromodulators such as acetylcholine, noradrenaline, and serotonin, thought to be involved in volume transmission, a form of communication where signals diffuse through the extracellular space, are produced by cell groups generally located just beneath the lining of the ventricles or the outer surface of the ventral brain. For example, the nucleus basalis of Maynard, containing most of the brain's cholinergic neurons, is located in the basal forebrain, close to the optic nerve and the wall of the lateral ventricle. The locus coeruleus, the main source of noradrenaline in the brain, is situated along the lateral wall of the fourth ventricle. The serotonergic dorsal raphe nucleus is centrally located in the brainstem, but sends extensive projections to the walls of the lateral, third, and fourth ventricles. While it remains to be definitively proven, the strategic location of these neuromodulator-secreting neurons or their projections suggests that these signaling molecules might be distributed via CSF transport after their release from nerve terminals. In contrast, the dopamine-producing neurons of the substantia nigra are located deeper within the midbrain and do not directly contact brain surfaces bordering the CSF. Similarly, their projections do not terminate at the ventricular surface. This might reflect a need for more localized and tightly regulated dopamine signaling or a mechanism to prevent widespread dopaminergic modulation driven by CSF. Perhaps the most compelling example of communication between the brain and CSF involves the hypothalamus. Certain nuclei within the hypothalamus densely innervate the pituitary gland, an endocrine organ located at the base of the brain. A key feature of the pituitary is its location outside the blood-brain barrier and the CSF-filled spaces of the central nervous system. The pituitary gland sits in a bony indentation at the base of the skull and is separated from the subarachnoid space by a dural membrane that prevents CSF from directly accessing the gland. As a result, pituitary hormones are secreted directly into the bloodstream, bypassing CSF transport within the central nervous system. The pituitary gland is divided into two functionally and developmentally distinct lobes. The posterior pituitary gland contains axonal projections from large neurosecretory neurons located in the supraoptic and paraventricular nuclei of the hypothalamus. These neurons produce oxytocin and AVP, which are stored in the posterior pituitary and then released into the blood. Importantly, these same magnocellular neurosecretory cells can also release oxytocin and AVP from their dendrites within the hypothalamus itself. Dendrites from the supraoptic nucleus form a network just below the surface of that nucleus, while dendrites from the paraventricular nucleus form a bundle near the third ventricle, suggesting that oxytocin and vasopressin are distributed within the central nervous system via CSF transport, in addition to their actions in the periphery. Cortical signaling by oxytocin and AVP can influence social and cognitive functions without relying on direct synaptic connections. The anatomical arrangement of the hypothalamus and posterior pituitary suggests that oxytocin and AVP could exert their effects on social behavior and cognition through a combination of central and peripheral actions, potentially involving the regulation of both vascular tone by blood-borne peptides and synaptic tone by interstitial fluid-mediated peptidergic signaling. In contrast, parvocellular hypothalamic neurons secrete neurohormones that are transported to the anterior pituitary via specialized portal blood vessels, thus avoiding CSF transport and distribution within the central nervous system. These neurohormones control the release of a range of anterior pituitary hormones that regulate growth, lactation, stress responses, reproduction, and metabolism throughout the body. The precise role of CSF flow in the complex interplay between the hypothalamus, pituitary gland, and the regulation of glymphatic flow is still being investigated. The activation of the adrenal gland in response to stress, a classic example of a hypothalamic pituitary cascade, triggers a coordinated, body-wide stress response. While chronic stress has been shown to impair glymphatic function, the effects of acute stress on brain fluid movement remain unknown. Sleep and circadian rhythms, fundamental biological states conserved across evolution, serve to restore bodily functions and anticipate changes, in the environment. Both sleep and the timing of our internal biological clock regulate glymphatic function. Most brain regions exhibit endogenous rhythms in neuronal excitability, with peaks occurring within a 4-hour window across the brain. This synchronized daily activity allows for complex behaviors to have performance peaks at appropriate times, such as learning and memory being enhanced during the active phase. Recent work has shown that, in addition to synchronized neuronal activity, the distribution of CSF between the brain and lymph nodes is also under circadian control, with increased glymphatic influx and clearance from the brain during the rest phase and increased lymphatic drainage during the active phase. Considering the rhythmic neuronal activity, the circadian cycle of glymphatic flow, and the hypothesis that synchronized neuronal activity can drive fluid flow, it's conceivable that fluid flow in the brain acts as a mediator of circadian entrainment. Individual brain oscillators might drive rhythmic fluid flow to the next oscillator in line, thus synchronizing brain activity across the 24-hour cycle. The hypothalamus, with its strategic location and high concentration of neuropeptides, is proposed as a key signaling area for bulk fluid flow. The suprachiasmatic nucleus, or SCN, of the hypothalamus is considered the master circadian pacemaker, synchronizing daily rhythms in gene expression, metabolism, physiology, and behavior. Classic studies showed that transplanting SCNs from rhythmic animals into the third ventricle of animals with damaged SCNs could restore behavioral rhythmicity, and this effect depended on a diffusible factor, suggesting that the SCN may directly communicate with the CSF to synchronize circadian rhythms. The SCN contains a distinct distribution of neuropeptides. VIP neurons, crucial for maintaining intrinsic rhythmicity, are located in the ventral part above the optic chiasm. AVP neurons, involved in regulating the flexibility of rhythms, are found near the third ventricle. Although both VIP and AVP are present in CSF, how they enter the CSF pool is not yet known. Given the vasoactive properties of these peptides, it's hypothesized that VIP released near the base of the brain could affect the vascular tone of the glymphatic system, while AVP released into the ventricles, upstream in the CSF pathway, might tune fluid flow in downstream areas. Whether AVP is released by the SCN into the ventricles via direct innervation, a small portion of tanocytes within the SCN, or another method remains an open question. One potential downstream target is the pineal gland, located near the center of the brain, highly vascularized, and bathed in CSF. CSF trace. The vascular system is essential for carrying oxygen, glucose, other nutrients, and immune cells throughout the body. The blood-brain barrier, while crucial for protecting the brain's delicate environment, also limits the transfer of many solutes between the blood and the brain. The brain obtains an ultrafiltrate of blood via the choroid plexus, a highly vascularized layer of cells lining the ventricles that is responsible for most CSF production. Therefore, the choroid plexus can be considered the starting point of the glymphatic system. In addition to transporting signaling molecules like growth factors from the blood into the brain, the choroid plexus contributes to brain homeostasis by producing and secreting various enzymes and growth factors into the CSF. Interestingly, the choroid plexus also expresses receptors for neurotransmitters and neuropeptides, including norepinephrine, melatonin, and AVP, which, upon activation, can modulate the rate of CSF production. How changes in CSF production rates affect glymphatic flow in the brain is still largely unknown due to technical limitations. Some studies suggest that conditions that reduce glymphatic function, like age and wakefulness, also suppress CSF production, while others have found no direct correlation between CSF production and glymphatic fluid transport. Thus, the interdependence of CSF production and glymphatic function remains unclear, but the choroid plexus is undoubtedly a key player in the feedback loop involving the brain, the glymphatic system, and CSF production from the blood. It's important to note that the blood-brain barrier's restriction of plasma ultrafiltrate influx is not uniform across all brain regions, particularly in the circumventricular organs of the hypothalamus, where the interstitial fluid is separated from the rest of the brain by defined glial boundaries. Furthermore, the blood-brain barrier does not completely prevent the regulated transport of ions, oxygen, molecules, and even cells between the blood and the brain parenchyma under normal and pathological conditions. While the blood-brain barrier is clearly vital for brain health, its precise interactions with glymphatic fluid flow are currently unknown. Bidirectional signaling exists between the vascular compartment and the brain. As mentioned, the vascular system uses the choroid plexus as a gateway for signaling molecules to enter the central nervous system. Conversely, the brain communicates directly with the vascular compartment via the posterior pituitary gland. Peptidergic neuronal projections in the posterior pituitary release neuropeptides like oxytocin and AVP directly into the bloodstream, affecting cardiovascular tone, kidney function, and other processes. Feedback from this peripheral pathway can influence cerebral blood flow at a broader level compared to more localized hypothalamic signaling, potentially altering perivascular space volume and thus impacting glymphatic flow depending on the brain state. The brain and the gastrointestinal tract engage in extensive communication, often referred to as the gut-brain axis. Given the glymphatic system's role in clearing metabolic waste from the brain, it is logical to consider its potential responsiveness to satiety and hunger signals. Intravenous administration of triglycerides can alter the lipid composition of CSF, and glymphatic transport can distribute lipids throughout the brain. Changes in the distribution of triglycerides between the blood, brain, and CSF are also associated with dysregulation of appetite. These findings support the idea that food-derived lipids and other hydrophobic molecules, after absorption in the gut and processing by the liver, can enter the CSF from the blood via the choroid plexus and across the blood-brain barrier. In the brain, the primary carrier of cholesterol is a apolipoprotein E, or APO, which is abundantly produced by astrocytes and the choroid plexus. This suggests that APO might be distributed in the brain via fluid movement in the perivascular space. Different isoforms of APO, when delivered via CSF, enter periarterial spaces with varying efficiency. Interestingly, APO genotype is the strongest known genetic risk factor for late-onset sporadic Alzheimer's disease, and glymphatic function is impaired in animal models of Alzheimer's. Therefore, the differential distribution of lipids derived from the gut along the perivascular spaces might directly contribute to brain pathology. The vagus nerve provides a direct neural connection between the gut and the brain. Upon feeding, visceral branches of the vagus nerve are activated by various neuroendocrine cues, including cholecystokinin and glucagon-like peptide 1, produced by enteroendocrine cells in the gut. Conversely, food deprivation can reduce the firing and retrograde signaling of the vagus nerve. Vagus nerve stimulation has been shown to increase glymphatic influx. Feeding itself might inherently enhance glymphatic function to promote the circulation of hypothalamic satiety signals within the brain. The gut peptide GLP-1 has also garnered significant attention in neurodegeneration research, and notably, treatment with a GLP-1 receptor agonist has been found to reduce CSF production. Similarly, fibroblast growth factor 21, or FGF21, is produced by the liver and, when injected into the CSF, can inhibit sugar and alcohol appetite via activation of a specific hypothalamic nucleus and regulation of the hypothalamic pituitary thyroid axis. Vagal nerve stimulation, GLP-1 signaling, and CSF-derived FGF21 regulation of the hypothalamic pituitary thyroid axis offer additional potential links between the gut, brain, and glymphatic function that extend beyond traditional signals like insulin, leptin, and glucagon. The microbiome, the vast community of bacteria residing in the digestive tract, is essential not only for digestion but can also influence neuroendocrine and inflammatory responses in the gut. The microbiome has been implicated in nearly every disease of the nervous system, although the underlying mechanisms are often unclear. There is some evidence that APO isoform can influence the composition of the microbiota. The microbiota can regulate factors like GLP-1, thus ultimately affecting vagus nerve activity. Furthermore, the microbiota's composition can somehow alter the tight junctions of the blood-brain barrier, potentially disrupting the carefully maintained composition of CSF and interstitial fluid regulated by the choroid plexus. While current evidence suggests a possible link between the microbiome and glymphatic function, the specific pathways, beyond vagal retrograde signaling or blood-brain barrier glymphatic interactions, remain elusive. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.